Protein folding process has been investigated intensively so that we pretty much understand many aspects, but still we need more studies. Okay, protein folding process is basically a spontaneous process I mentioned, but sometimes need extrinsic helpers because some process shows high energy barriers. There are three types in this category: PDI, protein disulfide isomerase; second, PPI, peptidyl prolyl cis trans isomerase; third, chaperone. First one will convert disulfide bond into just a sulfide bond or reverse reaction. Second type will convert cis trans isomerization of proline. Third will just make abnormally folded proteins into unfolded state. I'm going to explain the first one by using this protein. You are looking at the bovine pancreatic trypsin inhibitor, BPTI, that has three disulfide bonds, as you see here, between 14 and 38, and 30 and 51, 55 and 5. Okay, so schematically, the native forms with three disulfide bond. They started with arbitrarily unfolded and fully reduced, meaning the disulfide bond is reduced into cysteine, and then just let them fold in test tube, and traced what kind of the disulfide bond made during folding process. They found a native disulfide bond sometimes accumulate. For example, in this case, disulfide bond between 5 and 38, that is not the native disulfide bond, right? There is no any. This kind of abnormal disulfide bond containing intermediate cannot proceed to form native structure because abnormal disulfide bond just simply the block the further process. So this abnormal disulfide bond must be reduced and then let it goes, okay, for proper disulfide bond formation. In E. coli, DSBA, disulfide bridge forming enzyme, had been determined for each structure and having this kind of three domains, okay. Centered domain resembled the structure of a CO reduction. Actually, CO reduction has a function in catalyzing this kind of reactions. The protein with the disulfide bond could be reduced into two cysteine forms. Okay, so reverse reaction also possible. So this pink domain in this protein may catalyze this kind of disulfide bond formation and the breakage. second type, PPI, is related to the proline. First, we must understand stability of trans peptide bond, where you can see CO and NH position in opposite direction, so there is no steric hindrance. Okay, while in cis form, two CO and NHs, and also because of that, the two side chains are probably experience some steric hindrance, meaning what? Trans is much stable than cis, 100 times. I'm sorry, 1000 times more stable. Okay. However, in proline, as you see, side chains has this kind of chain. The last CH2 just to make a covalent bond with peptide bond nitrogen. So flipping this region in peptide bond, producing this one cis. So virtually two forms are identical. See, nitrogen just having identical shape. Slightly different in these regions, but basically due to this kind of similarity, transform and cis forms are virtually similar in their stability. Only 3 or 4 times more stable. That's why in real protein cis forms are actually sometimes present. But there is some problem. Two forms cannot be converted easily. Okay, so during the folding process, some proline exists in cis form by accident. They may be in trouble in making correct native forms where it must adopt strengths. Okay, so PPI just accelerate two forms. You are looking at the structure of PPI. PPI has another multiple names, such as rotamase, cyclophilin, cyclosporin A binding protein, things like that. It has 8 antiparallel beta barriers at the center, active sites with a green color with attached substrate position in one surface of one barrel. Third type, actually, chaperone. Cytoplasm of all cells contains folded proteins and folding polypeptide at high concentrations. Unfolded proteins with exposed hydrophobic patches aggregate easily by non-specific hydrophobic interactions. To overcome this problem, a class of proteins called chaperones have evolved to sequester unfolded polypeptides. There are many in this category, but we're gonna just look at GRO-EL and GRO-ES protein, which is kind of big size. And you are looking at the single polypeptide of GRO-EL, actually composed of 3 domains: apical, the beta at the center and alpha helices at both sides, and intermediate domain and equatorial domain, both with alpha helices. Seven of these make arranged in a ring with a symmetrical mode. They just stack each other with the equatorial domains positioned in at the interface. The cross section of these structures are shown here with the same colors. As you see, two layers, equatorial, just face each other, and two layers have some cavity in the middle. This slide shows two different functional states of GRO EL based on three-dimensional reconstitution, whether it binds to the another partner, GRO ES. Large conformational change of GRO EL occurs when GRO ES and ATP are bound. GRO ES molecule binds to the one of GRO EL ring and closes it of the central cavity. The GRO EL ring becomes larger and the cavity inside that part of the cylinder becomes wider compared to alone. We are looking at the structure of GRO ES. Each polypeptide has three domains with two extended portion. Okay, again, seven polypeptide. They just make a ring type with symmetrical mode. The green one, mostly hydrophobic residues, interact with GRO EL, and red one positioned on top of GRO EL and covering and closing the cavity. In this slide, you can see the possible functional cycle of the GRO EL and GRO ES molecule. First, an unfolded protein molecule, yellow color, binds to one end of GRO EL ADP complex with bound GRO ES green at the other end, at the other end. And the GRO ES is released from the transposition and rebound together with the ATP at the cis position of GRO EL. ATP hydrolysis occurs as the protein is folding or unfolding inside cavities. Okay. Next, ATP binding and hydrolysis is the in the transposition. Disposition is required for release of GRO ES and the protein molecule. This step. And finally, a new unfolded protein molecule can now bind to GRO EL and repeating another cycle.